Hello, my name is Sara and I'm going to talk about genetic engineering. What is genetic engineering? Genetic engineering refers to the direct manipulation of DNA to alter an organism's characteristics, phenotype, in a particular way. Genetic engineering, sometimes called genetic modification, is the process of altering the DNA in an organism's genome. This may mean changing one base pair, AT or CG, deleting a whole region of DNA, or introducing an additional copy of a gene. It may also mean extracting DNA from another organism's genome and combining it with the DNA of that individual. Genetic engineering is used by scientists to enhance or modify the characteristics of an individual organism. Genetic engineering can be applied to any organism, from a virus to a sheep. For example, genetic engineering can be used to produce plants that have a higher nutritional value or can tolerate exposure to herbicides. Now I'm going to talk about the types of genetic engineering. There are many, but I'm going to talk about three. Recombinant DNA. Recombinant DNA is the method of combining two different DNA molecule sets belonging to different species and inserting them into a host organism to achieve the desired genetic combination. Gene delivery. Gene delivery process is achieved by transferring a specific set of genetic material, DNA or RNA, from a foreign body to the body of the host cell in order to achieve the required gene expression. Gene editing. Gene editing involves the modification, restructuring, or alteration of the DNA in the genome of a selected organism. It is done in order to achieve changes in the physical traits of the living organism. How does genetic engineering work? To help explain the process of genetic engineering, I have taken the example of insulin, a protein that helps regulate the sugar levels in our blood. Normally, insulin is produced in the pancreas, but in people with diabetes type 1, there is a problem with insulin production. People with diabetes therefore have to inject insulin to control their blood sugar levels. Genetic engineering has been used to produce a type of insulin very similar to our own, from yeast and bacteria. This genetically modified insulin, Humulin, was licensed for human use in 1982. Now I'm going to talk about the genetic engineering process. This is not always like this because it depends on the method that you are going to use and the thing that you want to achieve. A small piece of circular DNA called a plasmid is extracted from the bacteria or yeast cell. A small section is then cut out of the circular plasmid by restriction enzymes that are like molecular scissors. The gene for human insulin is inserted into the gap in the plasmid. This plasmid is now genetically modified. The genetically modified plasmid is introduced into a new bacteria or yeast cell. This cell then divides rapidly and starts making insulin. To create large amounts of the cells, the genetically modified bacteria or yeast are grown in large fermentation vessels that contain all the nutrients they need. The more the cells divided, the more insulin is produced. When fermentation is complete, the mixture is filtered to release the insulin. The insulin is then purified and packaged into bottles and insulin pens for the distribution to patients with diabetes. And that's all. Thanks for your attention. I hope you like it.